Okay, everyone, so now I'm going to go through Unit 2 of kind of the consolidated version, maybe for you reviewing for the AP test. Unit 2, if we remember, is all about cell structure and function. Make sure my volume's up there. Okay, so things that you're going to really need to know here are kind of the organelles and how they contribute to the function of a cell. The structural features that we use to capture, store, and use energy. One of the major topics of biology is related to surface area to volume ratio and why it's so important. So that's another topic, and I guess content area to focus on. And that structure function. We have specialized structures to make everything more efficient. Okay, so quick review. Two types of cells, prokaryotes, eukaryotes. Pro means before, so karyo means nucleus. Therefore, prokaryote means before a nucleus. Those are things like bacteria and archaea. Eukaryotes means true nucleus, so those are the ones that do have one, including us. Plants, animals, fungi, and protists do. In terms of specific features outside of maybe just a nucleus, prokaryote versus eukaryote. Prokaryotes have DNA in a nucleoid, which is kind of a one circular piece of DNA that just kind of floats around in the cytosol. They only have ribosomes. And if you think about it, all structures need ribosomes in order to take that DNA and turn it into a protein, because ribosomes are the site of protein synthesis. So they just have ribosomes. They are typically very small and are considered primitive. Whereas eukaryotes, as we know, have tons of other organelles. We have membrane-bound ones, like the nucleus and the mitochondria and the chloroplast, and they have specialized functions. So because of all these additional organelles, they can be much larger in size and more complex. So what's the job of the nucleus? Just kind of going back, it's the control center, obviously, but that's where the DNA lives. It has a nuclear envelope surrounding it that is continuous with the ER. The ER is like the factory portion of the cell that can make and crank out proteins for travel outside of the cell. I'll get to that in a minute. There are pores that can control what enters and leaves, which is what allows, for example, mRNA out to go and make proteins later. Chromatin, I will talk about in a moment too. It's basically the spaghetti-like structure of DNA wrapped around histones, which are the proteins, and then they compile together to make chromosomes. The nucleolus is the nucleus of the nucleus, and that's where ribosomes are made. Here's the graphical picture of what that kind of looks like. So you can see the ER is continuous with the nuclear membrane, and you can see the pores, and so on. Ribosomes, their job is protein synthesis. They are found in all life. So they have both rRNA, which helps to form the ribosomal RNA, is what the little r stands for, form the structural component, and then the protein. It is composed of a large subunit and a small subunit. The exact size varies between specific organisms, but there's a large and a small. They can be located in two spots in the cell. There are the free-floating ones called free ribosomes, and their job is to make proteins the cell uses. And then there's ones that are attached to the ER, which gives the rough ER its name. And those ones are the ones that are made for export outside of the cell. The ER, rough, means that it has ribosomes on it, so their particular job is to package proteins that are made by the ribosome for secretion. So they transport it off to the Golgi, which is like the packaging mail center of the cell, and it also makes replacement membrane. The smooth ER doesn't have any ribosomes, so its job is not really to make proteins. It does other things like lipids, breaking down carbohydrates, detox, and so on. The Golgi ensures proper folding, basically makes sure everything is packaged up perfectly, It's got a cis face and a trans face. Just know that cis means same, trans means across. So when things are traveling, it goes to the cis face first and then off to the trans face. It packages things up in these tiny vesicles that can then bud up against the membrane and then be sent to export. Lysosomes, lyse, remember, means to break. So lysosome means to essentially digest things inside the cell. So it is a membrane, there we go, bound organelle that contains hydrolytic enzymes. It will break down food particles that are taken in by phagocytosis, or if it ingests other things, then it will break it down. The rupture or release of these enzymes into the cell also play a part in apoptosis. Vacuoles are like big storage containers for the cell. They can hold things like food, water, poisons if you need. They have membrane-bound vesicles, are also kind of considered a vacuole. So if you're packaging things for transport within the cell, that could be considered a vacuole. Plants, just to note, have a super giant one in the middle called a central vacuole that's really responsible for holding water and gives it its turgor pressure that kind of pushes on the cell wall to keep plants upright. So looking at transport, we go from the nucleus to the rough ER, out of the ER to the cis face of the Golgi in a vesicle, and then it moves its way through and comes out the trans face, and then it can actually export outside of the cell if it needs. The mitochondria, we learn a whole lot later about, is the powerhouse of the cell because that's where cellular respiration takes place, where we break down glucose and turn it into ATP. It has an inner and outer membrane. It is one of the organelles that's double membraned. If you remember the endosymbiotic theory, I think I touch on it later too, the idea that it was once its own living thing and that a cell came along and ate it, and then they lived happily ever after. Cristae are the specific folds. So for the mitochondria, that particular structure allows an increase in surface area. So if we tie it back to surface area is super important, you're basically increasing the availability of space to do cellular respiration and make more ATP. The matrix is the fluidy part in the middle. The other double membrane bound organelle is a chloroplast. This is where photosynthesis takes place. So you can see it's kind of been likened to Oreos, where the thylakoid discs are kind of individually Oreos stacked on top of each other in piles called grana. And there's kind of fluid floating all around. That's the stroma. And this is where chlorophyll lives, the pigment that absorbs sunlight to actually do photosynthesis. So tying it back to that endosymbiotic theory, the idea that once upon a time there was an ancestor eukaryote that had a nucleus and it engulfed a mitochondria. So therefore it was kind of then able to produce a lot of ATP. And then those would lead to the common cells like we have in us today. And then at least one cell then after that fact must have engulfed a chloroplast for the same purpose. And those became our photosynthetic eukaryotes or plants. And some of the evidence that suggests this is true is that they are double membraned. So they had one, and then when we engulfed it, it got a second one. They have their own ribosomes, suggesting they can make their own proteins, and they have their own DNA independent of the DNA found in the nucleus. They can also reproduce independently within the cell. So those are all key pieces of evidence for that endosymbiotic theory. Peroxisomes, think peroxide. They break down fatty acids. So kind of similar to a lysosome, except it is focused around using peroxide to break down fatty acids. So here's kind of this animal cell in a nutshell. You can even see also additional pieces kind of hanging off like a flagella which is like a super long tail. You might have cilia or tiny little hairs that could potentially also help with movement as well. Things that you would not find in an animal cell such as this, you won't find any chloroplasts. There's no central vacuole, just smaller, littler vacuoles. There's no cell wall. And there is no plasmodesmata, which are the kind of junctions between two adjacent plant cells. This is what that cell looks like. You can see it does have chloroplasts, it does have a cell wall, and at the bottom you can see these little adjacent connectors, the plasmodesmata. Plant cells do not have lysosomes, they have peroxisomes. They don't have centrioles, which is important for mitosis, which we'll talk about later, and they don't have flagella because they're not really moving around. Okay. Again, with the surface area, cells have to maintain a large surface area to volume ratio, and this really has to do with the transport of nutrients throughout the cell. If you had one supergiant cell, you really wouldn't be able to maintain the energy costs of keeping that cell alive. It would take too long to transport necessary things from one side of the cell to another. So it's more advantageous to be small and have a lot of membrane area to make ATP, to get things from one side of the cell to the other, to interact with the environment through the membrane, those kinds of things. Membranes also allow you to localize metabolic processes. So for example, the lysosomes, you don't want those digestive enzymes to be free-floating all around all the time. So you want to keep them contained. So it helps to compartmentalize certain reactions in specific locations within the cell. This just kind of reiterates that same point and highlights some of the things that are membrane bound. One just highlighting an example of surface area would be like your alveoli in your lungs. This one is another one with your intestines, kind of maximizing surface area with villi, if you ever have to come up with an example. For plants, they have things like root hairs that basically maximize water absorption surface area. Okay. So just kind of reviewing, you have these in your slides somewhere. It's a good table to go through. You can always pause the video and read through this. I'm just going to kind of give it a second and scroll through. All right, so now we're to the idea of the plasma membrane and permeability. So you just have to understand what's in a membrane, what the fluid mosaic model is, and how things actually go through the membrane and permeability and things like that. So the cell membrane, as you know or should know, is selectively permeable. It does not let anything and everything through. So there are rules that we will go over. Fluid mosaic model just means that the membrane is held together by weak interactions. So the components of the membrane itself can kind of move a little bit. There's some give, fluidity. Mosaic means that it's composed of a lot of different components, whether it's phospholipids, proteins, carbs, things like that. In terms of the bilayer, it is a bilayer, meaning that it has two layers, and you can kind of see it's made of two rows of phospholipids, making it amphipathic. That's a super fancy word for meaning that it has a hydrophilic portion and a hydrophobic portion. So the heads of the phospholipids really like water because they're polar, and the hydrophobic tails of the phospholipid are water-fearing. They are nonpolar, so they like to hide on the inside, and so they organize themselves in this way because the cell and outside the cell is basically a bag of water. Hydrophobic barriers allow and kind of keep out anything and everything from being able to move between the cells. Okay, so in terms of fluidity, saturation and cholesterol can affect that fluidity. So basically, if we remember back to saturated and unsaturated fatty acids, that applies to the tail. So if they're fat or if they're saturated, they're straight. If they're unsaturated, that means they have some double or triple bonds that make kinks, and therefore they're farther apart. So ones that are more tightly packed are going to be less fluid and therefore more likely solid at room temperature, which is why saturated fats are things like butter. If it's unsaturated, then they're farther apart, meaning it's more fluid, and that could be things like oil. Cholesterol can impact this because it can kind of stick itself in between those fatty acid tails, and it helps to minimize all the movement and fluidity that might occur at higher temperatures. As you heat something up, molecules start to move around a little bit more, so again, that makes things more fluid too. On the flip side, cholesterol can also help keep things spaced out when temperatures get low because things stop moving, so it can kind of prevent super tight compaction of the tails. So it acts as a buffer. There are two types of proteins really in the membrane. The integral ones are the ones that are truly stuck all the way through the membrane itself. They are transmembrane, so a lot of times as they're structurally organized, thinking of proteins in a 3D shape, on their outside they have hydrophilic heads because that's where the hydrophilic water area would be, and then the hydrophobic amino acids will kind of fold themselves toward the interior of those proteins. Peripheral just means like if your peripheral vision, it's on the side, it's on one particular side. So it's either on the extracellular side or the cytoplasmic side. It's not really embedded throughout. It's held in place by the cytoskeleton, which is like the skeleton component of a cell, and it just helps to add structure. Here's roughly what that looks like. So you can see the big fat ones going all the way through. Those are your peripheral ones. The integral ones are the integral ones are the ones that go through. Peripheral are the ones stuck on one side. And then you can even see the little cholesterol molecules. There might be other proteins attached to one side or the other. In this case, we have things like carbohydrates and glycolipids and other things. This is what one of those transmembrane or integral proteins might look like. Oftentimes there's the N terminus, which is kind of the start side on one side, and then the C terminus, which is the ending side on the other. The hydrophobic section is going to want to place itself in the middle, similar to the hydrophobic tails. And the hydrophilic ends are going to want to be towards the hydrophilic part on the inside and outside. These really come into play when we talk about channels and cell signaling later on in Unit 4. Some of the things that they do, they transport, they could act as enzymes. Proteins have all kinds of functions, as you know. In C, they might be a signal receptor, so kind of like a ligand binding to a receptor. We'll learn about that in Unit 4. It might be cell-to-cell recognition, or intracellular joining of somehow, or attachment purposes. There's a lot of functions. Carbohydrates usually are the ones that are involved in cell-to-cell recognition, thinking about things like our blood groups. And if you have type A blood, you have A antigens or glycoproteins on the surface of your red blood cells. The membrane is selectively permeable. And here are basically the rules. If it's small and non-polar, meaning that it's hydrophobic, it doesn't have a charge, because the interior of the membrane is also hydrophobic, they kind of can jump across really easily. They're small enough to get past the hydrophilic heads and tails, and they move on through. These include things like carbon dioxide, oxygen, nitrogen. Small polar molecules, as long as they're overall uncharged, so they might have some polarity, but they're not charged like ions, then they can move across. So for a small amount, we can move water across. For larger amounts, then you'll need something else, but small, it can happen. Now, hydrophobic core of the membrane really prevents charged and polar things from going on through. So ions can't go through, large polar molecules can't go through. You need specific transmembrane proteins to make that happen. If you think about plants, they have an additional cell wall, so they kind of have an additional hurdle to come through. And so they have a structural boundary similar to animal cells, but things can move through that as well. Okay, so now we're going to talk about transport and how things move. So you have to be able to know the different types of transport across a plasma membrane and the types of things that might affect it. Passive transport means that there's no energy required. So those include the topics of diffusion, osmosis, and facilitated diffusion. Active transport does require energy because you're moving things kind of against where they normally would go. And then the last category is bulk transport. So those are for really, really big things in terms of bringing in and out of the cell. So passive transport is how we do a lot of things, including gas exchange in our lungs. There's no energy because things are moving down a concentration gradient. It's going from where there's an area of high to low concentration, so a lot to a little. And then eventually, if you get the same amount on both sides, then it reaches a state of equilibrium. Osmosis is a special name that we give to the diffusion of water. It still is passive because water is essentially moving from where it is most concentrated to where it is less concentrated. So when the solute molecules are too big to actually do the moving itself, water moves. So for an example over here, there is a high concentration of water compared to over here. So what happens is there's a net movement of water kind of trying to equal out the concentrations of solute. So since the solute itself can't move, water moves so that you can equal out the concentrations. This brings about a couple of vocab terms: hypotonic, isotonic, and hypertonic. Hyper means excessive or too much. So if something is hypertonic, you have too much solute, or you have more solute compared to whatever the other solution is. If it's hypotonic, that means you have too little solute. And iso means the same. So depending on if something is hyper or hypo, if the solute molecules are too big to move, then that dictates where water would move. So for example, if you took a blood cell and put it in a hypotonic solution, meaning that there's less solute, water would rush into the cell to try to equal out the concentrations and therefore might cause it to burst. Whereas a hypertonic solution, the water would rush out of the blood cell trying to make the surrounding solution equal in concentration and then cause it to shrivel. When it's isotonic, it's equal on both parts. So what's entering and leaving is the same, and you have a nice, happy cell. Plants are a little bit different because they have cell walls. So they kind of somehow have some structural integrity. But they normally actually want to be in a hypotonic solution because they want to fill up with water that will then give them something called turgor pressure, which allows them to stand upright with those long stems and stalks. If it's just isotonic, it ends up kind of being a little bit flaccid. And if it's dehydrated enough, meaning it's in a hypertonic solution and water rushes out, it can shrivel kind of inside the cell wall. Facilitated diffusion is another passive transport. There's no energy required, but it does need a helper. It's facilitated. There's some kind of channel or carrier protein that helps these hydrophilic things go through. It's either a channel that basically just kind of opens up and allows them to move through from high concentration to low concentration when they can't pass through the membrane by themselves, or it can somehow loosely bind and kind of carry it across. So ions do this, other polar molecules, for example, water, if you're trying to move a lot of water, or glucose. The special protein channel that does it for water is called an aquaporin. It's a pore for water. It basically just opens up and allows it to flood through. Active transport, on the other hand, does require energy. So you're going to need ATP to make this happen. And really that's because you're pumping things against the concentration gradient. So from a low concentration to a high concentration. And there are specific pumps that make this happen. So there's a sodium-potassium pump in neurons. Whenever there is an action potential, then the pump kind of helps reset things. There's also the proton pump in the electron transport chain when we learn about cellular respiration. And this just kind of talks about those in picture format right here. So to kind of recap, passive has no energy. You are moving from high to low concentration down the gradient, and that includes diffusion, osmosis, and facilitated diffusion. On the right, you can see that it does require energy. You are moving things against the concentration gradient from low to high, and this includes pumps. It also includes endo and exocytosis, which we didn't really talk about, but kind of the bulk entry and exit of materials. Here's a picture of that recap. All right, so just knowing that we can't do it all the time everywhere, there is a regulation control piece involved. So a lot of organisms like this paramecium, for example, has something called a contractile vacuole. So it might fill and squeeze out water as a regulation measure to try to maintain a homeostasis. So that way the cell, or I guess the organism in this case, doesn't end up dying because it gets shriveled or explodes or so on. Here's just reiterating that bulk transport process. So endocytosis means taking in, exocytosis means releasing. Same process, just going in opposite directions. Depending on what you're doing, there's different names for it, but I think as long as you know endocytosis means taking in, you're good. Here's a graphical representation if you want to pause a second and kind of see the breakdown. All right. So at once upon a time in this section, we learned about something called water potential. It was an equation. I don't like it. You probably don't like it, but here it is. So water potential is basically a way for you to put a number on the likelihood of water moving from wherever it has a high potential to a low potential. Water potential is just the energy of water, so kind of the likelihood that it will move. We also have something called solute potential, which is roughly the solute concentration. And then there's pressure potential, which is kind of how much physical pressure is on the solution. Usually this comes into play with plants. We arbitrarily give it a 1. If it's an animal cell or in pure water, then usually it gets a 0 because it's so fluid that there really isn't any pressure. But overall, water potential is a combination of the solute and any pressure that might occur. Water will flow from wherever there's high potential, so a high likelihood of movement, to a low water potential, wherever there's a low likelihood of movement. Here's the general formula. So it is the solute potential is negative ICRT. So I is a measure of ionization. So it is the number of particles that it would make in solution. For example, if you have salt in water, salt is NaCl. So it will become, when it dissolves in water, Na and Cl. So its ionization constant is 2. If you have MgCl2, then that would eventually dissolve into 3 particles. C is the molar concentration, so moles per liter. Generally that's given to you, but you could potentially calculate that chemistry style. R is a pressure constant. It is always the same, so we would always use 0.0831, and that would be given to you. Temperature is in kelvins, so it's whatever the Celsius measure is plus 273. All of that unit conversion and this equation would be found on the equation sheet if you actually needed it. So also thinking if you were to add water, it's going to lower the solute potential. It makes it more negative because if you add water, or excuse me, let me backtrack. If you add solute, then you're going to increase the concentration. More solute means you're trying to dissolve more in the same amount of water. So that would increase the molar concentration. And therefore, when you add here or multiply, it would then be a negative. So by increasing any of these numbers, if you increase temperature, if you increase the amount of particles, if you increase concentration, it's all going to yield a higher negative. So by doing that, you are lowering the water potential, lowering the potential for water to move away from that location. So generally speaking, it's always going to move from a low solute to a high solute. It's going to move to the location where it can try to equalize concentrations. It will move from high pressure to low pressure as well. So just kind of getting an idea of where water would move. If there's pressure, it might move in one direction. If there's not, then it's going to move toward the area that has more solute. And really, this water potential is why water can move up a tree. So at the bottom, there's so much water compared to solutes that it has an extremely high water potential. And then as you go up, you can see this one has an extremely low water potential because there's not a lot of water, a ton of solute. So water is basically going to kind of be sucked up the tree because of that difference. And that was the kind of final example of this unit. So just get a general idea for the equation and what it in general means if you ever had to use it.